Hello again. This is part 3 in the Major Events in Evolution video series. And in the past two of these videos, I've talked somewhat about organisms, but more about the physical nature of Earth and the Moon and how that system led to life on Earth. So in this next video, part 3 here, I'll talk more about some of the biological aspects and some of the complex biological aspects that have occurred over the past 4 billion years. As I indicated in previous videos, the evolution of organisms isn't a simple bifurcating tree. As in these orange lines, that's the fairly simple bifurcating tree, similar to what Darwin first proposed back in the 1830s. But now we know that many different organisms have joined together. So organisms from the archaea, some from bacteria have joined together to form these initial eukaryotes, and then other protists, and then eventually plants. And there have been additional endosymbiotic events to form even more organelles. And here's that circular tree that shows the same thing. So you have bacteria, archaea, and eukaryotes, and then you have an archaeal line and a planktomycete coming together to form the initial eukaryotes, and then an alpha proteobacterium coming out of the bacterial line, joining with that initial eukaryotic cell to become a mitochondrial eukaryote. And then one line over here has gained a chloroplast from a cyanobacterial line here. And then there are some tiny lines in here, which I've accentuated in the next diagram. So if we blow up just that eukaryotic line, here's the nucleus coming in, mitochondria coming into this eukaryotic line, and then the plastids here to become the plants. But then there are several other multiple endosymbiotic events that led to this chromalveolate line, or now the Stromenopyles, alveolata, and rhizarial line, as well as some other protists as well. So on this diagram, you can see it a little more clearly. So this is the basic one, leading to the nucleus, mitochondria, chloroplasts, and a chromatophore in the rhizaria up here, which is another cyanobacterium entering a different set of protist cells to become the chromatophores in the rhizaria. Subsequently then, some red algal cells and some green algal cells became endosymbionts and eventually organelles in the rhizaria, in the chromista, which are the Stromenopyles, in the alveolata, and in this group, which are now the Hacrobia, and in the excavata. And there have been also endosymbiotic events of these cells then into the alveolates as well. So there have been primary endosymbiotic events, secondary endosymbiotic events to become organelles, and tertiary events as well, and possibly in some cases quaternary, that is, a cell within a cell within a cell within a cell. So it's known that these first endosymbiotic events to form a nucleus, that is a eukaryote, and then a mitochondrial eukaryote, occurred sometime between 2.2 and 2.4 billion years ago. And then the formation of the plants by taking in a cyanobacterium to become the plastid, that happened around 1.9 billion years ago. The endosymbiotic event that led to chromatophores in rhizaria here, between a protist cell and a cyanobacterium, probably took place somewhere between 1 and 1.2 billion years ago. And then all of these events here between red algae, green algae, and these various protists down here took place within the last billion years probably. And then these ones to form dinoflagellates in the alveolata probably took place more recently than that. Then the question arises, has this happened even before 2.4 billion years ago in the bacteria or archaea? And so to address this, people have looked at the gram-negative and gram-positive bacteria. Gram-positive bacteria are often called monoderms. They have only one membrane. And gram-negative bacteria are often called diderms because they have two membranes. So the question is, how did diderms obtain their second membrane? How do they make it? It takes a lot of genes to make a membrane and a lot of proteins that fit into it. So many genes are involved, and it's a complex system. So how does that happen? So a few years ago, a paper came out to address this, looking at genes in common among the gram-negative and gram-positive bacteria to determine whether an endosymbiosis between two gram-positive bacteria led to the gram-negative bacteria. And so that's the conclusion of that paper was that, yes, it's possible that there was one bacterium here in what's called the Chloroflexi phylum and one from the Firmicutes. If you put those together, then the genes that are found in the gram-negatives are agree with at least a combination of these two types of bacteria coming together as one. And so that sort of makes sense. Here are these monoderms here, including the Chloroflexi and the Firmicutes, and you join two of those together, and then you get this diderm group. However, this little branch here, these are the cyanobacteria, which are also diderms. Furthermore, they have a difference in their photosystem, so they always on almost every tree come out closer to monoderms than they do to diderms. And so a few years ago, actually more recently than that, just a year or so ago, I tried to address this question. So I won't go into all of the details here. The paper came out last year, and I will discuss it in a little more detail later in the semester. But this is a phylogram based on some endosymbiotic events. So down here, this basal group, those are the monoderms. So they have only one membrane, and then this dotted line is a separation between the monoderms and the diderms. And also shown in blue and red colors, and actually purple here, are the photosystems that they have. So there were early photosystems down here. They diverged into a type 1 photosystem and a type 2 photosystem, or RC is for reaction center 1 and reaction center 2. All of the other photosynthetic bacteria have either RC1, or reaction center 1, or reaction center 2. But cyanobacteria has both of them, RC1 and RC2, and those were actually the reaction centers then that became the ones used in chloroplasts because they come from cyanobacteria, right? So those are also the ones that you find in plants. And they diverged way back here. So several billion years ago, those two photosystems diverged from a common one here. But then they started to use different reaction centers that handle electrons differently. So they diverged. They're not the same. They don't have the same absorption spectra. That is, they don't react to the same wavelength of light. And yet you can find both of them being used in cyanobacteria in its duplex photosystem, using both photosystem 1 and photosystem 2. So how did those come together, and why is cyanobacteria so different? And the only thing that I could come up with when I analyzed the information that is available is that there was an endosymbiotic event here that formed the cyanobacteria with both photosystems, and each photosystem has between 60 and 100 and something genes responsible for those photosystems. And then in these other lines, each one of those bacterial phyla either lost both photosystems or lost one or the other. They simply weren't compatible in those organisms. And then there's a couple of other organisms, phyla, that don't quite fit in there, and they've always been troublesome to position on any phylogram. And it looks as though they're the result of a different endosymbiotic event, one that's similar to the paper that was described about 10 years ago as a possible endosymbiosis between two monoderm species to become diderms. This diagram shows it a little more simply. So there was this ancestral reaction center which evolved, and these 1, 2, 3, those are mutations that are involved going from one phylum to another, one species to another, essentially. And so you then have this reaction center 2 here and reaction center 1, which led to the cyanobacteria, which eventually became oxygenic. The first ones probably were not oxygenic. And then you have these other phyla of bacteria, some that lose both reaction centers and some that lose only reaction center 1 or reaction center 2. And so they specialize in absorbing one type of light only. And these gray lines over here indicate the second potential endosymbiotic event, where none of the species that came out of those endosymbioses were photosynthetic. But on this side then, these are all diderms and these are all monoderms on this side. So it appears endosymbiosis has been a major cause of some actually major evolutionary events on Earth, dating back into the bacteria and then continuing into creating the eukaryotes and various different groups within the eukaryotes. And remember, as I think I said in the last video, you're joining entire genomes together during these endosymbiotic events. And so there have to be genes and proteins that are compatible. And they don't have to be completely compatible, but they can't be detrimental to the organism. They have to be overall beneficial combinations of those genes and the proteins that they encode. So this is a little bit more simplified of a diagram. And so the major gram-negative bacteria up here appear to have been formed by joining of a photosynthetic member of the Firmicutes and a photosynthetic member of Chloroflexi. And those photosystems, again, had diverged from a progenote cell long before. There's a second event over here to lead to the minor groups of diderms or gram-negative bacteria. And that appears to have been between, again, a Firmicutes, either a photosynthetic or non-photosynthetic, because none of those gram-negative bacteria that we know of are photosynthetic, and an actinobacteria, and none of those members are photosynthetic either. So that leads to gram-negative bacteria, all of which are not photosynthetic at all. So here's a diagram that kind of summarizes all of those, and don't worry about getting all of the details at this time. I'll be showing this diagram in several different lectures. But you have down here this initial 1 or 2 endosymbiotic events, and every time I have a red and a blue line here, that indicates an endosymbiotic event to form the gram-negative bacteria. There are probably at least 2 of those. And then you have a eukaryotic progenote, which comes out of the archaeal line, joins with a planktomycete to become the eukaryotes with a nucleus. Those then have another endosymbiotic event between those amitochondrial or non-mitochondrial eukaryotes and an alpha proteobacterium, which then becomes a mitochondrion. And then one of those members joins with a cyanobacterium to become the photosynthetic eukaryotes, including glaucophytes, red algae, and green algae. And the green algae then diversify into mosses and ferns and gymnosperms, angiosperms, etc. There's another event over here to form this pollinella, which is a rhizarian. So there's a couple different events between cyanobacteria and eukaryotes. One forms the chloroplast and the other one forms chromatophores, which are analogous to chloroplasts. Next, then, within these protists, there are many other endosymbiotic events. Between a couple here, a green alga and a mitochondrial eukaryotic protozoan to become the excavata, and those diverge into a euglenoid group, which are photosynthetic, and then trypanosomes on this side, which are not photosynthetic. Although if you look into the trypanosome nucleus, look into its genome, it still has genes from the cyanobacteria that were for photosynthesis, but they've gone on to take on a parasitic lifestyle where they no longer need to make their own carbon, so they don't need photosynthesis. So that also indicates a sort of gain in function initially to become photosynthetic, and then a loss of function, although some of the genes are still retained and some of them are actually used, but not for photosynthesis. Then there's a group down here, which is an association of another green alga and a mitochondrial eukaryote to form this group, the Chlorarachnophyta. And then red algae here are taken up by mitochondrial eukaryotes to become ciliates, cryptomonads, haptophytes, heterocons, and these are the alveolata here, mainly dinoflagellates. So in most cases, the chloroplasts are retained, but not in all cases. And in some cases, that initial nucleus is still retained, and it's called a nucleomorph because it's shrunk quite a bit from its original source, either a red or a green alga. And as far as I know, none of them have retained the mitochondrion in the organelle, but the mitochondrion in the main cell is the one that's been retained. Then up here in the dinoflagellates, you can see now it's another endosymbiotic event between some of these cells and some of these cells to take on even more complexity. That is, again, it's a cell within a cell within a cell within a cell. So at least a tertiary endosymbiotic event, and in some cases possibly a quaternary endosymbiotic event. And again, remember, all of these are carrying a lot of DNA in the nucleus, in the mitochondrion, in the plastid, and then between two eukaryotes, it's the nucleus, chloroplast, and mitochondrial of the endosymbiont, plus the nucleus and the mitochondrion of the host cell. So there are a lot of genes being imported or mixed in each one of these events. So a lot of duplication of genes, and I'll talk about this later in some videos, how those sort out. They're not all used and not all retained in the long run. And it goes all the way back to bacteria, so it's been happening a very long time, and it's much more common than was what was once thought. There's even more complexity added because there are horizontal transfers of genes, not whole organisms, that have been happening also from the beginning of evolution on Earth. So horizontal gene transfer at its ultimate is the endosymbiosis of two organisms joining their genomes together. But other types of horizontal gene transfers occur so that a single gene can be transferred, or 5 genes can be transferred, or 10 genes can be transferred at a time into an organism, so that they can sort of trade different sequences back and forth. And this can happen by several means, and I'll talk about those in more detail in a different video. A few years ago, a group of researchers decided to look at the extent of horizontal gene transfer in a group of bacteria, and they took 181 genomes, that is the entire genome of each one of those bacteria, and started to compare them. This tree here was determined just by ribosomal RNA genes, and those are very well conserved genes, and they also aren't moving around. So these are ones that really are not moving around horizontally, but only vertically. That is, they're only inherited from the parent to the progeny. Next, they superimposed the ancient lateral or horizontal transfers that they could measure within these genomes. So you can see all of these horizontal transfers of genes that are moving between species from one to another. Next, they found common genes among other organisms at a higher taxonomic level. So these were ones that occurred more recently. And then they compared the genes that were in common between pairs of species. That is, so these would be very recent transfer events. So when you put all of those together, superimposed over this ribosomal RNA gene tree, you can see all of these gene transfers that have been occurring, some very long ago, some more recently, and some very recently. So they're constantly occurring, these transfers of genes, one at a time, two at a time, or several at a time, moving from species to species, and then being incorporated into those genomes. So this makes studying molecular evolution a lot more interesting, but much more difficult too, trying to trace the lineages and trying to trace not only the lineages of the species, but the lineages of the genes. So just a summary of some of those movements then, in the endosymbiotic case, in plants and animals, then you have DNA introduced by these cells moving into other cells, becoming endosymbionts, but then there's movement between those organelles of DNA, and there's also transfer of DNA from bacterium to bacterium, from archaeum to archaeum, and from bacteria to archaea, either directly into eukaryotic cells or through viruses as well. So there's DNA that can be introduced in many, many different ways in cellular organisms, as well as in viruses and through viruses. But the changes in DNA don't end there. So if you look up here, if you have two independent organisms, they tend to maintain their genome or even add sequences from the outside to their genome, but they don't change significantly over short periods of time. However, if you have a parasitic organism that is gaining some nutrients from a host organism, then normally the parasite loses DNA sequences because it doesn't need those genes, so they are free to mutate and eventually mutate out of existence, so the genomes tend to get smaller. In a mutualistic relationship where both partners are gaining from the other partner, then both genomes tend to decrease. So they're actually losing sequences. And then in an endosymbiotic relationship where there's also transfer, a lot of the genes from the endosymbiont are transferred into the main nuclear DNA. And so the nucleus can tend to get somewhat larger, although it loses some genes that are duplications also, and the genome of the endosymbiont and eventually the organelle becomes smaller and smaller. So if we go all the way back to the beginning here of this diagram, we essentially have bacteria first gram-positive or monoderms and then joining together to form diderms. So now you've joined two different organisms together, but there's also horizontal gene transfer going on. So at that time, it would look something like this, where you have transfers among bacteria and archaea, as well as among viruses and between viruses and those organisms. But then we start getting these eukaryotic organisms being formed, including those with a nucleus, those with a mitochondria, and eventually those with chloroplasts as well. So then you have these horizontal gene transfers and endosymbiotic events leading to more exchanges of DNA. And then there's transfer within each one of those organisms, so that you have transfer of DNA normally from the organelle into the nucleus. And so that each one of those organelles tends to lose DNA over time. And this occurs with the more complex organisms as well, as I already indicated. So you have this organelle now, which was a red or green alga. There have been transfers within those original organisms as well as in the organelle, and then transfer mainly to the host cell nucleus in this case. So those host cell nuclei contain DNA from a lot of different sources then. So here's a further expansion of that with, at first, an animal cell picking up sequences from bacteria, archaea, viruses, plant cells, dinoflagellates, and other protists. And then from the mitochondria, the mitochondrial sequences have moved into the main nucleus. In plants, similar idea here, except now you have an extra organelle here, the plastid, where some of those sequences wind up in the nucleus mainly. There's a few cases where some of those sequences may have moved into the mitochondrial genome also, and some sequences have moved from the mitochondrial genome into the plastid genome. Then in dinoflagellates and other protists, where you have a eukaryotic cell inside of a eukaryotic cell, then you can see all of these movements of DNA also, both from horizontal gene transfer and then these endosymbiotic events where you have DNA from a plastid, a mitochondrion, and a nucleus of the organelle that can move also around in that cell. So then evolution overall consists of this vertical bifurcating tree here. So there's inheritance vertically, but there have been many endosymbiotic events shown here, and this is not an exhaustive list of them, which have joined several genomes together, 1, 2, and sometimes more genomes together, into the same organism. And so there's been sorting out of those genes, and these are illustrated here on this diagram. So already it's become fairly much more complex than just a bifurcating tree. And then when you add on all these horizontal gene transfers or lateral gene transfers, then all of that becomes much, much more complex, more interesting, but it presents a very complex picture of evolution, especially at the molecular level. Okay, so that's the end of part 3 on major events in evolution. And as I said, in this one, I concentrated more on the organisms than on Earth history. Next time, I'll go into some detail about nucleic acids, and then we'll continue on discussing some of the important molecules and processes that DNA and RNA are capable of. So that's it for today. Talk to you later. Thanks very much.